Hi, everyone. My name is Erin Lashnits. I am a veterinarian and a clinical assistant professor at the School of Veterinary Medicine at the University of Wisconsin-Madison. I'm presenting this lecture for Invisible International's Education Initiative, and thank you to the Monte Calvo Family Foundation for supporting this effort. I don't have any super relevant disclosures other than all of the very generous grant funding that went into a lot of the work that I'm going to present. No unapproved or unlabeled uses. And then the only other disclaimer that I would make is that I'm a veterinarian, not a human doctor. And so I don't want anyone to take this as medical advice. Rather, I am presenting some of the results of research that I've done thus far in my career. So I have a few learning objectives here. First is I hope by the end of this presentation that you'll be able to list the pathogenic bacteria that are carried typically by cat fleas. Secondly, to describe the characteristics of cats that are associated with Bartonella infection and particularly Bartonella clarigiae infection in their fleas. And then thirdly, I hope that you'll be able to understand the role of Wolbachia species in cat fleas. As far as an outline, this kind of just will take you through a little bit of background on some of the flea-borne pathogens I'll be talking about, then the specific aims for the research paper that I'll be reviewing, for that study, the methods and results, and then some broader conclusions and future directions for this research. So in terms of background, the first thing I should introduce to you all is the cat flea. The cat flea is Stenotaphylides felis, so I'll probably just say cat flea. You'll see this abbreviated as C. felis as well. C. felis is the most common ectoparasite of domestic dogs and domestic cats worldwide. It's able to act as a vector for multiple zoonotic organisms. The main ones that we think about are two bacteria, so Bartonella species and Rickettsia species, and then a tapeworm, Diploidium caninum, which is the cat and dog tapeworm. When it comes to bacterial pathogen infection, which is what I'll be focused on here, I'm not going to talk anymore about the tapeworm. But when it comes to bacterial pathogen infection in cat fleas, we really don't have a good understanding of the factors that influence what pathogens infect different cat fleas, what percentage of cat fleas are infected, or the implications on risk for human infection. So what are the risk factors for human infection with these flea-borne diseases or animal infection as well? Again, I'll be focused on the human side, but for me as a veterinarian, I also focus on the animal side in my own work. A little bit of background on the cat flea. The cat flea does have a more complicated sort of phylogenetic and family history. So the cat flea is separated into 8 different what we call clades, and this is based on genetics, phylogenetics. These are geographically distinct and limited by mainly climate, but also sort of geography. So there are 2 temperate clades, and then 3, 4, 5, and 6 are the tropical clades. Cat fleas are more common in tropical locations, but they can be found worldwide, as you can see based on these maps. And I'll be talking about the different clades through the research that we've done as well. In Africa, there are 2 specific clades that are just specific to Africa. We do not find these typically in the US. So delving a little bit into the pathogens that can infect these fleas, the first one that I'll talk about a little bit is Bartonella. Bartonella species can be transmitted by cat fleas. And also there are specific Bartonella species that are associated particularly with cats themselves. These include Bartonella henselae, Bartonella clarigiae, and Bartonella cholerae. So you can see those written up on the PowerPoint. Sometimes they're abbreviated with just the letters, but mostly I'll be talking about Bartonella henselae and Bartonella clarigiae today. We know that all of these Bartonella species, as well as many others, and there are probably over 40 at this point that have been named, they can cause both human disease and disease in animals. And it's interesting because a few previous studies that have looked at the cat-hosted flea-transmitted Bartonella, so these 3 Bartonella species, they have actually showed poor agreement between the Bartonella species that infects the cat and the Bartonella that infects the cat's fleas. So two things we know. One is about the cats. The cats can maintain long-term and even asymptomatic infection of Bartonella, and that's the case with all of these cat-adapted species. But we also know that the fleas can maintain infection, and that has been shown for up to the lifetime of the flea, which in experimental studies is about 2 weeks. And so in these experimental studies, they've shown that that Bartonella can infect the flea gut and is passed in the feces for that entire period of time. So that may explain the discrepancy between some of what we're seeing in the cats and what we're seeing in the fleas. That said, even if cats are infected long term, they cannot transmit Bartonella species to one another in the absence of fleas. So again, experimental studies have showed this previously, where if you take a bunch of cats that all have Bartonella infection and you mix them together, they don't spread one to the other. However, if you add fleas to the mix, then they can all transmit. So we assume that the same is true for transmission of Bartonella species to humans from cats, that there's required to be a flea intermediary. And that mainly is because of the infectious material is the flea feces. So Bartonella is bacteremic in the cat's bloodstream. However, it's not often that we as humans come in contact with cat blood entering our bloodstream, but if they have fleas, then either the flea bite can inoculate or the flea feces can become inoculated under the skin when you scratch your flea bites, or when a cat scratches you, the same as it would be scratching itself with its flea bites. So that just shows sort of the importance of the flea as the vector, that it is required for transmission. All right, that all being known, we still don't totally understand what specific thing is the driver for the risk for human infection. We know that fleas are probably required. We know that cats are probably required, but we don't know whether it's the cat infection, the flea infection, or some combination of both that's driving the risk for human infection. So moving on from Bartonella to a couple of species that we know a little bit less about. So the flea-borne Rickettsia species, there are two main species that I'll talk about in the introduction, and then one other one that I'll talk a little bit more about when I get to the research itself. Cat fleas may in fact serve as a vector for Rickettsia typhi in certain hyperendemic regions, even in the US. But outside of those specific locations and even within those specific locations, Rickettsia typhi infection of cat fleas is pretty uncommon. So what are the species that do infect cat fleas? Mainly, these are the Rickettsia felis-like organisms. These are Rickettsia felis-like because of Rickettsia felis. That's kind of the prototypical one that infects cat fleas. But then there's also this Rickettsia called Rickettsia assembinensis, and another one that's a candidate species, Rickettsia senegalensis. And those are the ones that are more typically found in cat fleas. Rickettsia felis-like organisms can cause clinical signs in humans, typically sort of an acute non-specific illness, fever, rash, headache, nausea, muscle and joint pain, similar to what you might expect for Rickettsia typhi or spotted fever group Rickettsias, but seemingly a little bit less severe. These are pretty new to human medicine, and so, or newly discovered in human medicine, have probably been around for a long time. And so we just don't know as much about the clinical manifestations. Similar to Bartonella, but probably even more so for the flea-borne Rickettsias, we really don't understand what the risk factors for flea infection of Rickettsia species, much less human infection with these flea-borne Rickettsias are. So the third bacterial pathogen that I'll talk about didn't make the list up front because this is hemotropic mycoplasma, and these hemotropic mycoplasmas are pathogens of cats, although they are not typically zoonotic. So they're not typically the same species that are transmitted cat to cat, are not typically transmitted to humans. These are red blood cell parasites, previously known as either hemobartonellas or eperythrozoans. And the new species names, or the current species names, the main one in cats is Mycoplasma haemophilus. And then there are two candidate species that are thought to be maybe a little less pathogenic, Mycoplasma haemominutum and Mycoplasma turacensis. As I said, these are pathogenic in cats, but generally don't cause human infection. But these are the cause of feline infectious anemia. Typically it causes a little bit of a fever, but generally just anemia in cats. Occasionally, we will detect hemotropic mycoplasma in cat fleas. And this has been shown across a handful of papers that were happily reviewed just this year by one of my colleagues, Charlotte Moore. And so the cat flea, because of this, has been proposed as a potential vector for hematotropic mycoplasmas, but actually that's probably incorrect. In lab experiments, cat fleas really don't transmit hematotropic mycoplasmas efficiently, and it's more likely that this low level of detection that we see across lots of papers of hematotropic mycoplasmas in fleas is more evidence for hematotropic mycoplasma in the blood meal, so in the cat blood that's inside the flea, not that the flea is acting as a true vector that's required for transmission. So then finally, Wolbachia species. Wolbachia, as you may recognize from other arthropod-borne diseases, is a commensal of various arthropods. Broadly, Wolbachia species infect a majority of arthropods and also helminths. And they can have a huge effect on the vector itself. So they can affect reproduction of these vectors, the vector competence, the efficiency of transmission of pathogens. And that can depend on the Wolbachia strain and how it interacts with different bacteria, the microbiome, and the vector itself. There are 3 Wolbachia strains that have already been isolated from cat fleas, and they're listed here. But we really don't know what these different strains do, how they interact with the fleas themselves, or their effect on the vector or the pathogen transmission at all. So that brings me to the paper that I'll review today. This is some work that I worked on towards the end of my PhD and into the beginning of my job here at UW, and owes a debt of gratitude to Charlotte Moore, who is a current PhD student working at NC State. So the specific aims for this paper or this project were to determine the cat host and the flea vector factors that were independently associated with flea infection with Bartonella and Rickettsia. So we looked at a number of cat level factors, things like the sex of the cat, the weight of the cat, the geographic location the cat lived in, and the pathogen infection of the cat itself. We then also looked at flea factors, and that included the phylogenetic clade, as I reviewed those clades from a previous paper before, and then bacterial colonization with various pathogens or with the commensal Wolbachia. We had some specific hypotheses on the most common flea Bartonella we expected to find, and that it would be Bartonella clarigiae, since that's been most commonly found in fleas previously. We also thought that cat geographic location and flea clade would be independently associated with Bartonella clarigiae infection of fleas. And then finally, that fleas collected from cats infected with Bartonella clarigiae would be more likely to be infected with Bartonella clarigiae. This would make sense. Thirdly, we hypothesized that fleas co-infected with either Rickettsia or Wolbachia would be more likely to be infected with Bartonella clarigiae. Moving on to our methods. We collected fleas from a population of cats that were likely to have both fleas and pathogens. And so these were free roaming cats, community cats, stray cats, feral cats that were enrolled through trap, neuter, return programs. So these are programs that go out into the community, trap these cats that are living free in the world, and then neuter them, sometimes provide a little bit of medical care, in our case, flea treatments, and then send them back out to their communities. So they were enrolled at the time that they were trapped and anesthetized for their spay or neuter, and then left to go back to their lives. Right after they were put under anesthesia for their spay or neuter, the cats were combed using a standardized protocol to look for, count, and collect whatever fleas were on them. And then these fleas were all stored and shipped to NC State, where all of the testing was done. Fleas, once they arrived in NC State, were ID'd to the species level and then washed using our specific washing protocol and crushed for DNA extraction. DNA was then extracted from individual fleas. The fleas were not pooled in any way, and this is different than some previous studies where fleas are pooled by cat. We did not pool our fleas because we wanted to compare on an individual flea basis what we were seeing with the pathogens. And then we also, to compare pathogens in cats to the pathogens in the fleas, we collected the ear tips from the cats. This is a routine thing that's done for cats that are spayed or neutered through these trap, neuter, return programs. Is the very tip of their ear is surgically removed while they're under anesthesia for their spay or neuter to be able to visually identify that they've been spayed or neutered already, so that if they get trapped again, we don't bother collecting them and re-anesthetizing them. So we were able to collect those ear tips that were removed during surgery and test those for the pathogens as well. DNA was extracted from the cat tissues using pretty standard protocols. And so this is the list of the locations that we sampled, the number of cats, and the percentage of cats that we actually got fleas from. You can see the timeline spanned a pretty wide span. A lot of this was due to COVID and not being able to sample as much as we would have liked during some of these times. But we had a number of different locations and a fairly good sample of at least 25 to 30 cats from each location. When it comes to sequencing, we used quantitative PCR and followed with sequencing for Bartonella, Rickettsia, Wolbachia, and using the COX-1 gene for identifying the flea species phylogenetically as well. You can see all the primers and the references that we used here. Now we'll come to our results. Looking specifically at the fleas themselves, we collected from these cats over 90% cat fleas. That may not be shocking, but it is interesting because that means that there were some fleas that were not cat fleas. We also found that each individual cat only had one species of flea. So if we found a cat that did not have cat fleas, it didn't also have cat fleas. And here you can see a map of the different locations that we sampled and the different types of fleas that we found. The lighter green is the non-cat flea species, and those were actually only found in Madison, Wisconsin, not in the North Carolina or Virginia locations. This also shows the different clades, and you can see that clade 4 was most common down in the Southeast, whereas clade 1 was more common here in Madison. This is the list of the other flea species that we found on the cats in Madison. And it was a handful of different fleas, which is very interesting to see such diversity on our outdoor cats. So what pathogens did we find? I'll go through hemotropic mycoplasma quickly, since it is, again, not a zoonotic pathogen, but we did find it. So in terms of hemotropic mycoplasma, as expected, we found it in cats, in a fair proportion of cats, about 10% of cats, but we didn't ever find it in any fleas. So this is probably more evidence even that fleas are not a major vector for hemotropic mycoplasma transmission in cats. Even though we had some cats that were infected, we did not find it in any of their fleas. These cats are probably getting infected by other routes, and the most likely hypothesis is probably direct blood-to-blood or fighting contact. Moving on to our Rickettsia species. Rickettsia species, we sort of found the opposite for Rickettsia that we did for hemotropic mycoplasma. So for Rickettsia, we found Rickettsia in our fleas fairly commonly. About over 60% of the fleas were infected, but we really rarely found Rickettsia species in the cats themselves. Rickettsia sembenensis was the most common species by far that we found in fleas, but we did find some Rickettsia felis. Looking at Rickettsia sembenensis, since it was so common, it did appear to be geographically constrained, where it was found in fleas from North Carolina and Virginia, but it wasn't found at all in Wisconsin. So we do wonder if there is some geographic constraint, whether this has to do with the geography or the climate or what, we're not sure, but it did seem to be confined to those southeastern states. But we also found that each Rickettsia species was confined to a specific clade of cat flea. So Rickettsia sembenensis was actually only found in those clade 4 fleas in those geographic locations, whereas Rickettsia felis was found in both clade 1 and clade 6 fleas. So this may go some ways to explaining the geographic distribution or vice versa. Then moving on to our Bartonella species, which are a little bit more complicated, mainly because we found our Bartonella species fairly commonly in both cats and fleas. So you can see the numbers here. And we found Bartonella chloridae as expected to be the most common, but we also did find Bartonella henselae and Bartonella cholerae in both cats and fleas. The final species that's listed here, Bartonella alsatica, this is actually a rabbit Bartonella, and we found this in one of the rabbit fleas that we collected from one of the cats. Very interesting, has not been found before in cats or their fleas. So that begs the question, if we're finding Bartonella in cats and in fleas, are the same Bartonellas infecting the same cats and fleas? And you might be surprised to find out that the answer is no. So I'm going to put up the numbers to fill in this table here. And each of these is highlighted, so I'll draw your attention to the highlighted numbers first. The first number I'll draw your attention to is this 48 here. These are 48 samples in which both the fleas and the cats were negative. They didn't have any Bartonella. So those ones matched up. There were the predominance of fleas and cats that were both negative. But moving down that column here, you can see these cats where tissue samples from these cats all tested negative for Bartonella, but at least one of their fleas that was removed from their cat, that same cat, tested positive for one of these Bartonella species. Bartonella chloridae being the most common, with 10 fleas from negative cats testing positive for Bartonella chloridae. And again, I think this points out that we know that fleas can maintain infection for their lifetime, and so they may have Bartonella chloridae within the flea that hasn't yet transmitted to the cat. The opposite was also true, where we found some fleas that were negative for Bartonellas that were feeding on cats that were positive. So it may be that the blood meal hadn't transmitted yet. It may be just that there's low level bacteremia, and whatever blood that flea had taken up didn't happen to have the Bartonella in it at that time. But we found both cats with Bartonella chloridae and Bartonella henselae that had negative fleas on them. And then I'll point out that these are the ones that are concordant. So 4 cats with fleas that had both the cat and the flea had Bartonella chloridae. There were also some that had different species. So a cat may have had Bartonella chloridae where the flea had Bartonella henselae. Interesting, but not very common. And so looking at this table, it seems that it isn't necessarily a good predictor of whether the flea is going to be infected just to know whether the cat is infected. So what is associated with Bartonella infection in our fleas? We thought maybe like Rickettsia sembenensis or Rickettsia species, that Bartonella would be associated with flea clade. This was not the case. So you can see that the different Bartonella species are distributed across various of the clades. Clade 4 had the biggest diversity of Bartonella species. It was also the number of the clade that we had the biggest representation from. So we would expect that if we were able to sample more clade 1 or clade 6, we might find some of those others, but with such small sample sizes, we weren't surprised to see only Bartonella chloridae, since it was our most common Bartonella. So maybe geography plays a role. Could geography be playing a role in predicting what fleas are infected with which Bartonellas? There were some geographic patterns, and you can look more closely at this, this map and this table in the paper, but there were some geographic patterns, but overall, Bartonella species was not associated with flea clade or geographic location. So we ultimately were able to make a statistical model that looked at what was associated out of our various predictors that we initially included. And what we found was that only 2 cat factors were statistically associated with Bartonella chloridae infection in fleas. And that was Bartonella chloridae was more common in fleas that came from male cats than female cats, and more common in cats that were smaller compared to cats that were bigger. Neither of these has very easy or plausible biological explanations. And so we wonder if this is just a sort of sampling artifact from this sample, or whether there could be some biological explanation that we just don't totally understand yet. And again, we found that Bartonella chloridae infection in fleas was not associated with geographic location, flea clade, cat infection status, or infection of the flea itself with either Rickettsia or Wolbachia. So broadly, I'm going to put up a very complicated chart, which again, you can look at the paper to delve into more detail on this, but this goes through for each pathogen and for each sort of predictor, comparison between the flea and the cat. So I'll draw your attention to column B here. This is for the Bartonella species. On the very left-hand side, you'll see the boxes either green or orange. That indicates the cat was infected with either Bartonella cleridiae or Bartonella henselae. And then on the right is the number of fleas that were collected from that individual cat, and then a bar graph showing the number of fleas that were infected with each individual Bartonella species. And so you know, if these things were associated, you would expect to see all of the orange boxes on the left have orange bar graphs on the right, and all of the green boxes on the left have green bar graphs on the right. And you can see just visually looking at this, like that is not the case. So these things just aren't tracking together. And if you delve into each of these predictors, you'll see that none of them really work out that way. So it is kind of a mixed bag, and not strong associations with any of these things that we looked at. So what conclusions can we draw from this? One major conclusion was that Rickettsia sempenensis and Bartonella cleridiae were the most common pathogenic bacteria in fleas from free roaming cats in these geographic locations. We also found that hemotropic mycoplasma were not found in our fleas in this study, despite cats being infected at sort of expected rates. So we don't know if this is a geographic or methodologic bias that we're missing hemotropic mycoplasma, but more likely it's just that fleas are not acting as a major vector of hemotropic mycoplasma in cats. And our broader conclusions, sort of in general for flea-borne disease and cats, is that risk factors for infection with flea-borne bacterial pathogens in people, which is what we most care about from the human health side, they still remain not too well characterized. We know that there's things beyond just purely cat ownership or just purely flea infestation. And so we are still continuing to look at things like geography, certain characteristics of the cats themselves, the smaller cats, the male cats. Do they actually have some reason that these cats are more likely to be spreading pathogens to, specifically Bartonella cleridiae, to their fleas? Or maybe it's something more about the behavior around the cats, people's behavior with cats that drive those risk factors. Regardless, it is important to know what fleas are in that patient's exposure area, so what they could possibly even be exposed to, and what pathogens they carry. Rickettsia sempenensis, Bartonella cleridiae, certainly very common in our fleas in our sample set. Some of the other things may have more geographic limitations. It's also important to remember that cats are the reservoir species for some species of Bartonella, including Cleridiae, Henselae, and Coelerae, and those are found in the fleas on cats pretty commonly. Cats, though, don't always exhibit symptoms, and so seemingly healthy cats can be carrying Bartonella, Rickettsia, and can have fleas that are carrying those pathogens as well. And then finally, we do need to investigate more into the specific pathophysiology of both Bartonella and Rickettsia species and how they cause disease in humans so that we can understand what it looks like when people are exposed. In terms of future directions for my own research, I'm hoping to expand the geographic range of surveillance for cats and cat fleas. I'm doing this with something that I've called the FleaNet project. This is a multi-year study trying to basically collect fleas and test for pathogens from cats from all 48 continental United States. So we're trying to expand the geographic span of this project really immensely. If you're interested in learning more, I do have a website looking at that shows the current status, what fleas we've collected, what states we've collected from, and just some more information generally about fleas and flea-borne disease. The other direction that I'm hoping to take and continue to take in my research is understanding the risks for and the symptomatology and sort of pathogenesis of human exposure to these pathogens. So not just what are the risk factors for Bartonella or Rickettsia from fleas and cats, but what does it look like when people actually get infected, and how can we mitigate some of those risks? With that, I will close with my contact slide. Feel free to reach out with any questions that you may have about fleas or flea-borne disease or zoonotic disease from cats in general. And thank you very much for your time and attention.